Hi, my name is Pyrat. Today I would like to talk about how does the ribosome work. First of all, I will explain transcription, translation. Finally, I will explain working of ribosome. DNA contains data for produce protein. There are a few steps for produce protein from DNA. First is DNA transcription and translation. Transcription is the first step in many stages of gene expression using DNA, where the specific part of DNA are copied to RNA. In this, we call it mRNA. Translation is a process in which the ribosome in the cytoplasm or ER synthesize the protein after process of decoding DNA to RNA in the nucleus. The whole process is called a gene expression. Three factors are required in this process, including with mRNA, tRNA, and rRNA. mRNA, or messenger RNA, is a RNA that produced from transcription step. Transfer RNA, or tRNA, are molecular bridges that connect mRNA codon to the amino acid they encoded. rRNA or ribosome has main function in produce protein in living cells. Ribosome can be found in two forms in the cells. First is free form. The second one is binding with endoplasmic reticulum. We call it rough endoplasmic reticulum. This organelle binding with mRNA, which is a product from transcription state, and the other factor is tRNA or transfer RNA. It binding for growing protein chain. Ribosome has three slots for tRNA, the A site, P site, and E site. tRNA move through this site from A to P to E as they deliver amino acid during translation. Type of ribosome is depend on the type of living cell that they living in. First is eukaryote, including with 40S and 60S, and the other one is prokaryote, is including with 30S and 50S, and we call it 70S ribosome. Translation step has three parts. First, initiation, elongation, and the last one is termination. Initiation. In this stage, the ribosome gets together with the mRNA and the first tRNA, so translation can begin. Elongation. In this stage, amino acids are brought to the ribosome by tRNA and linked together to form a protein chain. The last stage is termination. In this stage, the finished polypeptide will be released into cell and do its job. This figure shows the whole process of the translation. First, tRNA will binding to the start codon, and this translation will start from 5' to 3', and it will stop when it sees the stop codon. tRNA will read the code from mRNA and bring the protein connect together for making a polypeptide chain. I hope you guys found this video helpful. Thank you for watching.